What will it take to seal the deal? Report by Andy Groves, PhD. Hearing Health Magazine, Winter 2014 edition. Hearing Restoration Project scientists have identified mammalian inner ear hair cells that take the first steps toward regeneration and then mysteriously stop. Here is how they will figure out why. This is the fifth article in a series about current projects underway in the Hearing Restoration Project. This piece explains transcriptional profiling of purified supporting cells from control and damaged adult mouse utricles, with and without inactivation of Notch signaling, the project of HRP Consortium members Andy Groves, Ph.D., Baylor College of Medicine, Neil Sedgwill, Ph.D., University of Southern California, and Jennifer Stone, Ph.D., University of Washington. We sincerely thank Dr. Groves for contributing this story to the magazine. In previous hearing health articles in this series about the Hearing Restoration Project HRP, we have discussed the problem of hair cell regeneration that in mammals, hair cells, the highly sensitive sound detectors of the inner ear, are unable to grow back after damage to the ear. As a result, hearing loss in humans is progressive and permanent. However, although humans and other mammals cannot replace their lost hair cells, this is not true for other vertebrates. It has been known for more than 25 years that birds, frogs, and fish can regenerate their hair cells naturally. A bird that has been deafened is able to grow back almost its entire complement of hair cells and hear again almost perfectly in a matter of weeks. However, mammals apparently lost the ability to regenerate hair cells at some point over the past 300 million years. How are birds able to regenerate their hair cells? Every bird hair cell is surrounded by 4 to 8 supporting cells to form a repeating mosaic pattern. The death of a bird hair cell somehow triggers one of its neighboring supporting cells to divide to give 2 daughter cells. One of the daughter cells then turns into a hair cell, and this process of division followed by transformation into a hair cell restores the normal mosaic of hair cells and supporting cells. Regeneration of this type happens normally in some parts of our bodies. For example, we lose and replace about 10 billion cells every day from the lining of our digestive system. However, although humans and other mammals also have supporting cells surrounding their hair cells, there is almost no regeneration of hair cells after damage in our inner ears. Some of the HRP-supported research has focused on the cochlea of newborn mice, which have a capacity for hair cell regeneration, albeit a rather limited one that disappears before mice start to hear at about 14 days after birth. Some HRP Consortium scientists are trying to understand the genetic changes that occur in the cochlea during this very brief time period. More recently, the lab of HRP member Jennifer Stone, Ph.D., at the University of Washington, has made an exciting discovery in another part of the inner ear called the utricle. The utricle also contains hair cells like the cochlea, but it uses them to detect gravity rather than sound. Dr. Stone and her colleagues have pioneered the technique of isolating the utricle from an adult mouse and growing it in a dish in the lab. Since the adult cochlea is too delicate to isolate and grow in a dish, this technique is the only one that allows experimental access to hair cells and supporting cells in adult mammals. The lab found that within a few days after hair cells are killed in the utricle, the surrounding supporting cells take the very first genetic steps to activate the program to make hair cells, but then they stop before the hair cells actually form. It is as though the supporting cells have received a signal to regenerate new hair cells, but they cannot circumflex seal the dihela and complete the regeneration program. This situation is very different from the cochlea, where absolutely no hair cell regeneration steps occur in adults. Can we therefore use the utricle to learn more about the signals that drive regeneration? In an attempt to identify factors that can push supporting cells over this roadblock, Dr. Stone turned to drug inhibitors of the Notch signaling pathway. In our report What Stops the Inner Ear from Regenerating? In the fall 2013 issue of Hearing Health, available www. At hhf.org, we discussed how notch signaling is an evolutionarily ancient form of cell communication that is frequently used to create mosaic patterns of different cell types, such as the mosaic repeating pattern of hair cells and supporting cells in the inner ear. In this scheme, supporting cells make a protein on their cell surfaces. This is the notch receptor. Hair cells make proteins on their cell surfaces that bind to the notch receptor, like a key fitting into a lock. Binding to the notch receptor is believed to actively block supporting cells from turning into hair cells. Dr. Stone and her team found that if they killed hair cells in the utricle, but then used drugs to block notch signaling, they saw small but significant numbers of true hair cells appearing in their experiments. These results suggest that the notch pathway may be one of the signals that holds regeneration back in the adult mammal. Neil Sedgwill, PhD, and I have teamed up with Dr. Stone and her lab to identify other players that may block regeneration in the utricle. Using Dr. Stone's culture system, our 3 labs will use next-generation DNA sequencing technology to identify genes that are switched on in supporting cells of the utricle as they attempt to transform themselves into hair cells after damage but ultimately fail. We will then see what genes are switched on or off when drugs that block the notch pathway are used to finally complete the process and drive supporting cells to produce hair cells. These experiments are still at a very early stage, but we hope this approach will complement other work by members of the HRP consortium that are looking at similar processes in the cochlea.